Hello. Quite early on in your GCSE you may be given the topic of chemistry of food. So you need to learn about the constituents of food and how enzymes break them down. Now a lot of the concepts here might be quite foreign to you, and I think it's easier to understand what's going on if you have a bit more detail and then simplify it from there. Statements that may be made are such as these: carbohydrates are made of sugars, proteins made of amino acids, and so on. You may learn about proteins being denatured for the first time. Enzymes as catalysts may also be new, and then that these digestive enzymes break down large molecules into smaller ones. If you're not aware of the smaller molecules to start with, that can be quite daunting a topic to learn. So let's start off with something very simple. You will have heard of simple molecules like water and carbon dioxide. Okay, molecules are just atoms stuck together, chemically joined in patterns. Now here's another small molecule, but the interesting thing about this one is its little pattern of molecules with a carbon, 2 oxygens and a hydrogen that I've circled here. We call that an acid group. So any molecule that has that combination of atoms in that order is termed as an acid. Now there are lots of organic molecules in the body. Organic molecules are just molecules that tend to be found in living things. Here are 2 examples. Glucose, again you may have heard of that, and adenine is part of DNA. Here are some different molecules. There's an amino acid on the left and a fatty acid on the right. You'll notice that they've both got those acid groups. That's why they're called acids, but they're very different molecules. Now amino acids, there are about 20 different ones of those, come in all shapes and sizes, but they all have that common feature of the acid group. In addition, they also have something called an amino group, and that's how they get their name. Now what happens in biology is that amino acids can be linked together. The blue amino group can link to the red acid group. Now if you've ever played the game called Barrel Monkeys, you'll know that you get little sets of monkeys and you can hang them and link them together. So we can kind of imagine a protein as a series of these monkeys linked with their arms, where the blue arm links to the red arm. And you can end up with a long chain, perhaps even 1,000 amino acids linked to make a protein. In the same way, fatty acids and glycerol can link together to form larger molecules that we call lipids. And again, in the same way, small sugar molecules like glucose and fructose can link together to form larger sugar molecules. It's all the same pattern. Now proteins are made from amino acids and sugars linked together to form carbohydrates. You can get links between proteins and sugars and proteins and fatty acids, but let's not make it more complicated. In fact, let's forget everything and keep it simple. What have we got? We've got that simple sugars make complex carbohydrates by being linked together. Fatty acids and glycerol can link together to make a lipid, and the amino acids can link together to make a protein. Okay, ready for the next bit of information. Proteins, and we only worry about proteins for this, can be folded into 3-dimensional shapes. The bonds that hold that 3-dimensional shape are quite weak, and therefore if we have high temperatures or extremes of conditions like 2 acid or 2 alkali for that particular protein, it will unfold, and we call that process denaturing. We say that the protein is denatured. Now the nutrients from your food are absorbed in the small intestine. They pass through the cells in the lining of the intestine and into the blood vessels and carried by the blood. Only small molecules can do this. Amino acids, simple sugars, fatty acids and glycerol can. Large proteins, lipids and carbohydrates cannot. So what breaks these large molecules down? Well, here are 2 starch molecules. If you put starch in water and heat it, The particles move around, and there will occasionally be collisions between starch molecules which break the molecules up into smaller ones. Over time, that will break down starch into individual glucose molecules, but that will take a huge amount of time and a high temperature. Enzymes allow reactions like this to happen at a lower temperature. They help the reaction to happen. Now let's use the example of amylase, which is an enzyme that, just because of its shape, helps break down starch molecules into small glucose molecules. The glucose and starch molecules fit into an area that is just the right shape, which happens to be called the active site. It's where the active part of the event happens. So here comes the starch molecule, and it's going to collide with an enzyme, not at high speed in an 80 or 90 degree liquid, but at body temperature. The collision is not that forceful enough to break it on its own, but let's look closely at what happens. The glucose units of the starch are attracted to parts of this active site. Once one of those glucose units is inserted in the active site, a second one tries to fit into the active site, but it's not quite the right shape, so it stretches the bond becomes under strain. Once the bond breaks, then this release of energy catapults the results of the breakage out of the enzyme, and the enzyme is then free to take another bit of starch and do the same thing. Now enzymes like this can do thousands of reactions, typically 100 reactions per second for several hours. Protein then might need replacing or repair. Try typing a 2-letter word 100 times per second for several hours. You'll realise how fast some of these reactions happen. Okay, let's forget all that again and concentrate on what we need to be able to say. Enzymes help a reaction happen. They catalyse it or speed it up, and they work on a specific reaction. They're specific because the thing that fits into the active site is unique. We call that a substrate fitting in the active site. Each enzyme has a specific substrate. Enzymes have optimum conditions. They work very, very fast under perfect conditions, but if you vary those conditions outside of the optimum, they will not work so fast, so their action rate will fall. And if you have extreme conditions, because they're a protein, they will denature, which means their 3D shape will unfold. Now you can watch some other videos that are specific for the details of optimum conditions for enzymes. For the moment, hopefully, we've turned you from a confused person to somebody that understands a little bit more about what we're talking about with this topic.